So good morning and welcome everyone. For today's session, we have Dr. Jeff Dong. Jeff is a postdoctoral fellow in Dr. Wee Young's laboratory. He completed his PhD in microbiology and immunology from the University of British Columbia. He is interested in understanding the function of tissue-resident macrophages in health and disease. And today he's going to talk about neurodegeneration and neuroprotection in MS. And in the audience, I see a lot of people who are new. So if you have any questions, you can post your question in the chat box and the speaker will address it once he finishes. So over to you, Jeff.
Thanks, Nitu. So good morning, everyone. Today, I'd like to tell you about neurodegeneration and neuroprotection in MS. So some of you may already be familiar with this picture where on the x-axis, it shows that the progression of MS over time and disability on the y-axis. When a patient first becomes diagnosed with a disability that's related to MS, we tend to call it clinically isolated syndrome. And this is seen when the disability is above a certain clinical threshold. If there are multiple recurrences of relapse remission or recurring disability, this is the relapse remitting phase of MS. And we tend to think that the relapse remission is driven by episodes of inflammatory relapses as seen by the blue bars. But it's possible that these inflammatory relapses have also occurred prior to the appearance of clinical disability. It's just that it's undetected because it's lower than the clinical threshold. Over time, as the disease progresses, you get progressive loss of brain volume and increase in axon loss. And when it reaches a certain point, the disability is no longer, you're no longer able to recover from disability. And this is when the patient enters the secondary progressive phase of disease where you have continual worsening of disability. There's also a small proportion of MS patients that are diagnosed as primary progressive disease where they do not undergo a relapse remitting phase. Much of the pathology which drives MS disability tends to be demyelination, inflammation, as well as degeneration that's occurring in both the spinal cord as well as in the brain, white matter, and cortexes. While we don't really know what's the initial cause of demyelination and inflammation in MS, we know that the vision loss, motor dysfunction, fatigue and depression, as well as other disability that's a result of relapse remission or demyelination and inflammation in MS can potentially be reversible. And this is along with processes that lead to inflammatory resolution, remyelination, at least partial remyelination, as well as CNS plasticity. So, however, while some of the disability can be reversible, a recent study showed that if you were to look at brain volume loss over time, and this is up to 13 years, in different RRMS patients, whether they have stable disease, whether they have non-worsening relapses, or they have worsening relapses, the rate of brain volume loss seems to be pretty consistent. So this means that despite of relapse and remission and activity or repair, that there is a consistent degenerative process that's occurring in the background, or in this study, they call it a silent degenerative process. Similarly, if you were to look at the survival of MS patients over a number of years, that despite them having different rates or different numbers of relapses, over, whether they have 1 to 2, 2 to 4, or greater than 5, the survival curve is very similar, again, suggesting that the progression of MS is independent of the number of relapses. So this could mean that there is a process of neurodegeneration that's independent of relapse remission, that's independent of inflammation and demyelination, and this is a process that's continuously occurring throughout the relapse remitting phase of disease as well as throughout progressive disease, and perhaps started as soon as the earliest points of diagnosis or even before that. So we really have to consider neurodegeneration as an aspect of MS pathology in addition to demyelination and inflammation. And we think that the disability that's caused by neurodegeneration is much more difficult to stop and reverse in comparisons to demyelination, where you can have remyelination, or inflammation, where you can have the resolution of inflammation. So if we go back and look at this particular picture, where we think that a certain clinical threshold, if you were to reach this point, you get progressive disease, it's also possible to interpret as if you were to reach a certain threshold of beyond the threshold of the capacity in which your neurons or your brain can handle neuronal injury, you start getting disability and disease. So really, the neuron injury capacity may be a factor in terms of how neurodegeneration occurs and leads to disability. So what does it look like in the MS brain? If you were to look at a MRI image of a patient where these images were taken at 2 months interval, the leftmost image shows the formation of a new MS lesion. So this is a gadolinium positive lesion that's due to demyelination, inflammation, and permeability of the blood-brain barrier, as you can see here. It's a white circle at the beginning of this particular relapse, but over time, 2, 4, 6 months, we see that the white or the gadolinium lesion disappears, but it leaves behind a dark spot. So this is an atrophy site that doesn't seem to recover. So despite that, the demyelination or the inflammation can resolve itself, the lesion or the atrophy site does not recover. And if we were to look longer term throughout the progression of MS, we can see that there's both brain atrophy, as seen here, where in normal patients versus relapse remitting MS patient versus progressive MS, you can see that the ventricle in the brain, the size of the ventricle dramatically increases. And this is an indication that there's brain atrophy occurring. Similarly, if you look at axon density in the spinal cord, the MS patient in progressive MS would have much lower density of axons compared to normal axons. Again, showing that there's a neurodegenerative process that leads to both loss of brain volume as well as axon density. What does this look like microscopically? So if you were to look at micrograph pictures of MS lesions, for example, you can see that there are a lot of these black bulbs. And these bulbs are the degenerating axons, both in the center of active lesions as well as occurring on the edge of MS lesions. You can also see them degenerating in the sense that the axons have transected into multiple pieces, as well as you have these swelling of the the axon bulbs. And together, this is termed Wallerian degeneration. With immunofluorescence, we can start seeing more details with these degenerating axons. So on the left here is staining of axons or fluorescent axons from MS lesion. And you can see that the green axons have these swelling bulbs. And the swelling bulbs, if you were to co-label with myelin, you can see that they don't have myelin around them, which is stained in red. And there are areas of demyelination that's also occurring. And you can see there's areas that's in the process of demyelination, areas that have completely demyelinated, and areas where the demyelination has led to this swelling of axons and formation of the axon bulb. We can also look at immune cells or phagocytes that's associated with the demyelinating axons. So here you can see that the macrophages and microglia in the MS lesion is labeled in red, and they are in close association with the green axons, especially the axons that have degenerated. Whereas on the right, you can see that these normal looking microglia are not really touching any of the normal axons, and they're only sampling or associating with the axon bulbs. And if we were to quantify the amount or the number of these transected or degenerating axons in active MS lesions or in chronic MS lesions, their numbers are significantly higher than normal white matter in the MS patient or in control white matter. So again, suggesting that there's this significant process of neurodegeneration and axon damage that's occurring in MS lesions. And the previous example I showed you are axon degeneration while there is demyelination. But there are also forms of axon degeneration called focal axon degeneration that could occur without demyelination. And again, this is a more recent study published in Nature Medicine, where they looked at MS lesions, and they were characterizing the loss of or degenerating axons into three processes or three stages, where stage 0 is on relatively normal axons, but stage 1 is when the axon swelling is starting to begin, and stage 2 is where you have transection. And you can see quantified here in these bar graph is that in MS lesions, you start getting significant increases in the number of axons that's in stage 1 swelling, and stage 2 where it's being transected. And looking on with these immunofluorescent images, you can see that the myelin that's in the orange, labeled by the orange fluorescence, is maintained, despite that in stage 1 and stage 2, you don't really have the myelin loss as seen in the previous pictures. And it's suggesting that you can have myelin degeneration in the absence of demyelination. We'll get into a little bit in how this occurs a little bit later. So what are the mechanisms that can lead to neurodegeneration in MS? We know that in our MS, this mechanisms or this neurodegenerative process is somewhat understood because we have relatively good animal models such as EAE in rodents that can help us model the inflammatory aspect of RRMS. However, we don't really have a perfect or any good model to model MS progression or the progressive phase of MS. So keep in mind that a lot of what I'll show you in terms of mechanism is largely due to what we understand with rodents, and it's more relative to relapse remitting MS rather than progressive MS. So in relapse remitting MS, we know that there's a significant infiltration of immune cells. For example, T cells that are autoreactive can enter the CNS, where they can help to activate resident glial cells such as microglia, astrocytes, or macrophages, or have the ability to directly activate neurons. But in terms of the overall response is that you have the secretion or the increasing levels of cytokines or dangerous molecules such as reactive oxygen species or reactive nitrogen species that causes stress on the neurons and axons. And when you have chronic inflammation that occurs over time, for example, when you have multiple relapses or when you have prolonged relapse, you start to initiate processes that can lead to CNS damage. So for example, oxidative stress, demyelination, ion channel imbalance, all of these together can lead to axon damage. And even though this particular figure shows them in somewhat of a sequential event, it's possible that they also affect each other, where, for example, if you have death from oxidative stress, they can directly lead and feed back into other processes. And we'll look at some specific examples, and we'll start with oxidative stress. So in this particular experiment, the authors used in the EAE model of relapse remitting MS. So you inject T cells, reactive T cells, or you immunize a rodent, in this case a mouse, with a myelin peptide, which causes T cells to become autoreactive. You can see that in the spinal cord, the EAE mice have significant increased amounts of the magenta colored macrophage microglia labeling, as well as the orange dots, which is something they've used to label for nitric oxide. So there's increasing amounts of reactive nitrogen species with increasing or accumulating macrophage microglia in the EAE spinal cord lesions. And if you were to directly add nitric oxide to the spinal cord, so this is live imaging of spinal cord after adding nitric oxide, you can see that over time you have transection of the axons, and that's quantified here. So this is suggesting that the reactive nitrogen species that's potentially being produced by activated macrophages and microglia in EAE lesions can directly lead to axon loss. Similarly, reactive oxygen species can also lead to axon damage. And here it's shown that, again, with the macrophages and microglia in EAE lesions showing purple, if you look at this particular assay, the AMPLUX assay, which detects hydrogen peroxide, we see that in comparison to the control, which is dark, the spinal cord in the EAE mice have a significant label, or is significantly positive for the H2O2 reaction. And again, when you in culture or in vivo addition of hydrogen peroxide to the spinal cord. So essentially, you open up the spinal cord and you subject the spinal cord to hydrogen peroxide. You can see this swelling and transection of axons, similarly to the treatment with the nitric oxide. And what's really interesting is that there's also mitochondrial defects. So you can see that outside of the lesion, the mitochondria is long and narrow, whereas once in the EAE lesion, where there's tons of the reactive oxygen species and reactive nitrogen species, the mitochondria are shorter and more rounded. So together, this suggests that the reactive oxidative stress can promote not only axon degeneration, but also mitochondria dysfunction. In our own lab, we also see the deposition of oxidized phospholipids in MS lesions. And this has been for a long time associated with oxidative stress in MS brains. But we also see that if you were to inject a form of oxidized lipids into the mouse spinal cord, you get the formation of these demyelinating and degenerating lesions, suggesting, again, these oxidized phospholipids have a contributing role to demyelination, neurodegeneration in MS. So we see that oxidative stress in the form of reactive oxygen species or reactive nitrogen species or oxidized lipids can promote damage not only directly to axons, but also perhaps indirectly to mitochondria, where then the mitochondrial dysfunction can lead to cell death. So together then, this is one aspect in which inflammation and the oxidative stress caused by inflammation can lead to axon damage. So now we'll get into a little bit about ion balance. So we know that when action potential occurs in an axon, it is together with a depolarization where you get the influx of sodium ions into the axon, and with a repolarization phase where the potassium ion is pumped out. So normally, the reason, or one of the main reasons that myelin helps with the conduction of the action potential, or the saltatory conduction, is that it allows this ion exchange to only occur at a small portion of the axon. So you only need ion exchange at these nodes of Ranvier where you don't have myelin for the action potential to occur, for the ion exchange to occur, and the signal can be transducted through the myelinated areas without much trouble. However, when you have demyelination, the unmyelinated axon don't have the ability to undergo saltatory conduction. So you need ion exchange along the entire length of the axon in order for the action potential to be propagated. So this is a problem because now you need much more sodium to be pumped out into the axon, and this leads to the problem of ion imbalance. And we can see this in MS lesions. So in the top, this is an image of the control spinal cord, or the green MBP labels for myelin, and the red labels for a sodium channel. And you can see that in control spinal cords, the sodium channels are localized to these small focal areas where there's very little myelin, and whereas you don't see the red labeling in the myelinated areas. In contrast, in MS spinal cord, where there's massive demyelination, you see that the red labeling is all throughout the axons, suggesting that not only there is a redistribution of the sodium channels, there's also likely an increase in the expression of sodium channels. And again, and this is to ensure that you do have the propagation of action potential, despite the fact that you don't have myelination for the most efficient types of action potential propagation. But this is a problem because once the sodium gets into the axon, it needs to exit the axon in order for the action potential to continue. And this happens when you have, in demyelination, because you have sodium, so much sodium entering into the axon, the cell needs to use alternative mechanisms in order to pump the sodium out. And this is where it engages these voltage-gated calcium channels in reverse sodium-calcium exchange, meaning that in order to pump the sodium out, the cell brings additional calcium into the axon. And this exchange leads to the accumulation of cellular or intra-axonal calcium. And this is a problem because too much calcium in the cell leads to the activation of casphases, which mediators of cell death, and this is what leads to injury and axon damage. In addition to the ion imbalance, glutamate may also be a problem. So glutamate normally is a neurotransmitter, and at low levels, it can help promote axon survival and regulate axon functions. But when you have neuroinflammation and you have the entrance of multiple types of immune cells as well as activation of astrocytes, they can produce excess amounts of glutamate. And it's thought that in addition to these pro-inflammatory functions of glutamate, glutamate itself can bind to AMPA receptors or NMDA receptors on axons. And the result of this is that you get additional calcium flooding into the cell. So not only you get calcium from the sodium imbalance, you also get calcium from increased glutamate levels. So together, this can lead to excitotoxicity. But the reason I put a question mark here is because again, glutamate has dual functions in that it could also be pro-survival. So it's not entirely clear whether the increased amount of glutamate in MS is beneficial or is it harmful, and we'll talk a little bit about that later when we talk about neuroprotection. But regardless, an increased amounts of calcium in the cell from multiple processes is bad. And that's summarized here, where the ion exchange is disrupted in the demyelinated axon, where you have accumulating calcium due to the sodium calcium reverse exchange, as well as potentially due to the glutamate receptor being activated. And this together leads to cell swelling, as well as mechanisms or processes that can lead to axon death. What about energy deficiency? So, like I mentioned, a normal myelinated axon only needs this action potential to be propagated through ion exchange at the nodes of Ranvier. Where when you have demyelinated axon, this action potential needs to occur throughout, or the ion exchange needs to occur throughout the entire length of the axon. And because these ion exchange channels, so the sodium pumps or the potassium pumps, all run on ATP, when you have the increased demands to run these channels, you get this increased demands of energy. So not only is the action potential much slower in the demyelinated axon, the energy demands is much higher. And this is a huge problem because more recent studies have shown that the oligodendrocytes and the myelin that's formed by the oligodendrocyte not only provides this ability to conduct action potentials, they also have an important function in providing nutrients to the axons. So oligodendrocytes, for example, can directly acquire glucose from the bloodstream, where once converted into lactate, can transport into the axons via these monocarboxylate transporters. And the lactate can then feed into the TCA cycle in the axons in the mitochondria. So in a model where you inhibit this process, for example, if you were to knock down or inhibit the expression of the monocarboxylate transporter 1, you get axon injury as seen here and quantified here. So this is suggesting that in demyelinated axons, you don't have, or you lose the capacity to provide nutrients to axons, and this can lead to decreased axon health. So together then with oxidative stress, with ion imbalance, with the loss of energy, proper energy needs, the demyelinated axon have a lot of problems, and it can start to degenerate. And if it's degenerating towards the soma or the cell body, we call it retrograde degeneration. And if it's degenerating away from the cell body, we call it anterograde degeneration. So in summary, neurodegeneration begins early in MS and continue with progression until the terminal phase of disease. In RMS, at least, the chronic or persistent inflammation and demyelination are the drivers of neurodegeneration, but we really don't know much about that in secondary progressive MS or PPMS or primary progressive MS, because we don't really know what drives our degeneration or the continual process of degeneration, as we don't have great animal models for these types of MS. And the processes that lead to degeneration in due to chronic inflammation and demyelination are processes such as oxidative stress, ion imbalance, and increased energy demands. So how can we protect axons from these degenerative processes? If you look at this schematic from a relatively recent review published in New England Journal of Medicine, most of the therapies that we have for MS are targeting the peripheral immune response or by inhibiting or depleting immune cells such as B cells. So while this is very effective for treating the relapse remitting phase of the disease because it inhibits inflammation and immune cell migration into the CNS, there's not a lot we can do in terms of neuroprotection, in that we can't really stop the degenerating and the progressive phase of MS. And this is also in part due to the fact that we don't really understand the processes or what caused these degenerative processes in progressive MS. So there's a need to develop new drugs and new therapies to treat progressive MS and neurodegeneration. And the current efforts have really been met with challenges and varying degrees of success. So like I mentioned, oxidative stress is a factor that leads to neurodegeneration. So perhaps we can try to increase antioxidants. And one of the successful treatments for relapse remitting MS, dimethyl fumarate, is a drug that can target reduce oxidative stress by upregulating antioxidant responses. So it's been shown to reduce annual relapse rate as well as reduce disease progression. And in part, this is by impairing immune cell activation, but more importantly, dimethyl fumarate has been shown to activate or upregulate the function of NRF2. And this is a transcription factor that, when activated and in the nucleus, can lead to the expression of multiple antioxidant proteins and anti-inflammatory genes, as well as cell survival pathways that overall can promote neuroprotection. So this is a successful example. But we really don't have a drug that can directly target or neutralize oxidative stress, and that's something that we are testing in our lab. We can also try to block sodium voltage gated channels, and this is to help with the imbalance of ions that I previously talked about. So in rodent models, for example, in rats, this somewhat has worked. So in this particular model of chronic EAE, where the vehicle, you can see a significant portion of the spinal cord having degenerating axons, and that's prevented by this drug called lamotrigine, which is essentially a blocker for sodium channels. And you can prevent the stress or the degeneration caused in EAE in rats. However, in clinical trials, when patients were treated with this lamotrigine drug, they actually show no overall benefit. And if anything, there's somewhat of a increased or detrimental effect throughout the period of treatments. So this drug was deemed unsuccessful, and perhaps not surprising, because even though sodium imbalance occurs in demyelinating axons, that's a result of the fact that you need to propagate the action potential. So if you were to use a sodium channel blocker, you might impair that entire process. What about glutamate excitotoxicity? Can we reduce the damage caused by glutamate? So this is a somewhat older study done in 2000, published in Nature, and this is where the idea that you can have the excitotoxicity caused by glutamate is where in EAE mice, they injected an antagonist against the AMPA receptor, the MBQX, and after injection, you can see that the severity of EAE clinical score is lower, suggesting that if you block AMPA signaling, you can reduce perhaps disease score or neurodegeneration caused by glutamate. And in a more recent study where in mice, they used a knockout of a subunit of the AMPA receptor. In EAE, you can also see that, so in here, the green is showing myelin, MBP, and SMI312 shows neurons or axons. Again, you can see that in the spinal cord, there's much less demyelination and there's much less neuron damage or axon damage in the AMPA receptor deficient mice compared to the normal mice or wild-type mice in EAE. Again, suggesting that inhibiting AMPA receptor signaling may be beneficial in preventing degeneration. However, one candidate drug, Riluzole, which was used as an AMPA receptor in trials, was not successful. So there was no difference between patients that were treated with or without this drug. So perhaps again, even though this particular mechanism or this drug works in animal models, it's a different story when it comes to human patients. But all the ion imbalance and glutamate excitotoxicity really comes down to the fact that you have intracellular calcium accumulation. So can we just block calcium voltage-gated channels to prevent that buildup of calcium? Maybe. So nimodipine, which is a drug that has been approved for blood pressure, and this is a blocker of a calcium channel, has been used in recent animal models. So for example, in EAE, if you were to treat the animal with the drug following peak of disease, you get much better recovery compared to if you don't treat. Similarly, if you were to measure the disability level, and this is by the paralysis or whether they can walk with the hind limbs, you can see that the treatment, both single dose and chronic dosing of this drug, was able to significantly reduce the amount of paralysis. Again, suggesting that if you block calcium channel or block calcium accumulation, you can reduce the severity of degeneration or disability in at least the animal models. And this hasn't yet been tested with MS patients, and I couldn't find clinical trials, so we'll have to wait to see what this drug can do in people. And since demyelination overall is a problem that both leads to ion imbalance as well as increased energy demands, can we promote remyelination then as an approach to prevent neurodegeneration? And this is, again, this is something that's newer in development. And for example, this one particular drug called clemestin, so this has been recently discovered or recently used in MS models, where here they show that if you were to inject mice with EAE with clemestin, you have reduced clinical score or reduced severity of EAE. And this is shown as a result of less axon demyelination as well as reduced axon loss. So again, it seems that if you were to promote remyelination, you can help reduce axon loss. And this is thought to act through as an antagonist to the muscarinic acetylcholine receptor on oligodendrocytes. So it's a promoter of remyelination. So this is currently in clinical trials with some promising results, but again, it's still limited at this point. And there's been some intense discussion around the use of exercise in helping mitigate MS disability, and this is perhaps due to the ability of exercise able to prevent or to promote neuroplasticity, to promote the activity of neural stem cells, and in our lab we show that exercise can promote remyelination. So perhaps exercise is another aspect, and it's a relatively easy and cheap way to prevent neurodegeneration in MS. And in addition to the beneficial effects I just talked about, we can also have upregulation of pro or neuroprotective neurotrophic factors as a result of exercise. So for example, brain-derived neurotrophic factor is something that's known to promote neural survival through the activation of PI3 kinase and AKT pathway. And if you were to take away BDNF, so in a BDNF knockout mice, if you take away BDNF and subject the mice to EAE, they actually have a much severe clinical scores for EAE. So this is suggesting that there's some baseline levels of BDNF even in EAE that prevents the severe worsening of the disease. And perhaps if we can then increase the BDNF levels above the baseline level, you can get better protection. With exercise, it's been shown that when in patients that have exercise, it's been shown that they have increased levels of BDNF in their serum compared to MS patients that do not exercise. So perhaps then this is a way in which exercise can help engage in a neuroprotective process, where not only it promotes plasticity, promotes remyelination, promotes neural stem cells, it also upregulates these neurotrophic factors that together overall can enhance neuroprotection. So going back to this particular picture where we have demyelination in the white matter, the cortexes in relapse remitting disease, where we then, when we move to progressive disease, you have these, you start getting atrophy, you start getting increased neurodegeneration, you get gray matter damage. This process is something that we still have trouble targeting, even though we have tons of disease-modifying therapies that can prevent the relapse-remitting disease. So by understanding the processes of neurodegeneration, particularly understanding how it happens in progressive disease, hopefully one day in the future, we will be able to have therapies that can also target the progressive phase of disease. So in summary here then, we see that most approved therapeutics for MS are good at inhibiting immune cells and inflammation, and by inhibiting this process, it's indirectly neuroprotective, but it doesn't really do much to the consistent neurodegeneration that occurs over long-term MS disease and over MS progression. And most of the drugs that we have tested with neuroprotective abilities tend to work in animal models, but they don't translate well into MS patients. And this can be due to a variety of reasons. And the progression in MS has been difficult to stop, and this is due to the lack of effective treatments, and probably in which that we can't really prevent or reverse neurodegeneration. So with that, thank you for your attention, and I'll look at the questions. So Peter asks, do we know for sure that the difference between RRMS and primary progressive MS is different in the underlying mechanisms, or is just a different manifestation of symptoms? So when an MS patient is diagnosed as primary progressive MS, it basically means that they don't have recovery. So they show disability, and the disability persists or continues to get worse. Whereas in relapse remitting MS, you get a episode of disability and it goes away and you recover. So that's the clinical manifestation, and that's how you diagnose them differently. But in terms of what causes primary progressive MS, we really don't know. And this is particularly because we don't have an animal model for primary progressive MS, and we don't know what causes this disability. And we think that perhaps it is just a neurodegenerative disease, where in relapse remitting MS, the inflammation and the demyelination is something that contributes to the neurodegeneration, but the neurodegeneration is something that has already happened, and is something that shows up or upon diagnosis more obviously in primary progressive MS. And perhaps in primary progressive MS, you just have undetectable levels of neurodegeneration and demyelination, and it shows, and it reaches a certain threshold, and it shows up in the clinics, and now you diagnose. So we don't know, I guess the simpler answer is. Ben asks, since there seems to be underlying progression in relapse remitting MS, would successful relapse remitting treatments targeting relapses simply result in progressive MS? So this seems to be the case, where a lot of the drugs where we can successfully prevent relapses, it doesn't stop them. So it can delay the onset of progression, but it doesn't stop progression. Eventually, as of current treatments, they will all most of MS patients will progress into the progressive phase. So yes, they will result in progressive MS, but the process can be delayed and the quality of life can be improved with these treatments. Wee says, whether primary progressive MS and relapse MS are different pathophysiology has been debated. Those support different pathophysiologies has brought up relapse remitting drugs being ineffective in primary progressive MS, and support that same disease but different manifestations brings up genetics in both forms. So there are some common elements of neuroinflammation, and all forms are primary progressive MS where there's axon degeneration that are not amenable to treatment, but there are extra lymphocyte infiltration in relapse remitting MS where relapses, extra lymphocyte infiltration in relapse remitting MS, which the relapse can occur because the lymphocyte infiltration promotes inflammation and they're easy to treat. That's a long comment.
You want me to clarify that further?
Sure.
Yeah, so that, that's a great question, right? The fact is, we still debate today if RRMS and PPMS are different diseases, and they're both chems. Usually one of the first things that people would bring up is that many of the RRMS drugs that we have don't treat PPMS, so therefore there must be different diseases. So that's one argument for them being different. But in terms of those that support the idea that they are the same condition with different manifestations, have brought in studies of genetic susceptibility where there does not seem to be any discrimination between the genetics of RRMS versus PPMS. And I mentioned this review that Peter Stice had written in Nature Reviews Neuroscience, maybe about 7-8 years ago now, where he shows that all forms of MS is a continuing straight line of neurodegeneration, that the underlying problem in MS is one of neurodegeneration. Not sure what initiates that, but neurodegeneration for everybody, a steep line that is not amenable, but that in those with relapsing remitting MS, there is superimposed inflammation that when they are substantial enough and get across the blood-brain barrier, they result in a relapse. So therefore, you do see the relapsing remitting form that is amendable to our anti-inflammatory agents, but basically the underlying progression that dictates all forms of MS is still not amenable to treatment. And that also helps explain your earlier slides that relapses don't really factor in the cause of progression. But it's a great question, still debated.
So Emily asks if exercise work in part by regulating BDNF, is it possible to supplement BDNF to obtain the benefit of exercise? So when reading the literature, there seems to be some caution about using BDNF as a treatment. So I'm not really sure whether it's good to supplement BDNF, and I'm not sure it's been tried. But I don't think in terms of being able to replace the entire benefits of exercise, it obviously can, because it's only one of the factors. So if possible, I think exercise is still the best way to go. And there are ways in which MS patients can exercise, and if they're more fatigued, for example, there are the sitting running wheels where you don't necessarily have to engage in normal walking, but you can still move your limbs, and that's a form of exercise. So yeah, I think exercise is still better than pharmacologically supplementing a particular factor. Sarah asks, do axon with demyelination versus axon with demyelination, so I think you mean do axons with myelin versus axons without, axons with demyelination versus without demyelination elicit different effects on MS lesions. For example, does one type perhaps worsen neurodegeneration? That's a good question. I don't know whether they have different effects in MS lesions. In terms of relapse remitting MS or in terms of the animal models of MS, it seems that, so demyelination occurs as a result of oligodendrocyte loss, whereas the axon damaged in the absence of demyelination is more perhaps acutely due to reactive or oxidative stress being induced by the immune cells. So it's possible that the axons that are lost due to reactive oxygen species are somewhat permanent, so they don't recover as well. Whereas when you have demyelination, it's possible that the axons are still somewhat alive, and when you have remyelination occurring, you can restore some of that function. So if going by that reasoning, it's possible that without demyelination, the axon loss could be worse, but I don't think that's directly compared. Hedwig says, considering oxidative stress and mitochondrial dysfunction go hand in hand, can you elaborate? So we have to think of those as something that occurs in parallel, where the oxidative stress is something that it's forced upon a cell by the environment. So when activated microglia or macrophages, when they make reactive oxygen species or reactive nitrogen species, it's normally against infections or against pathogens, but it's not specific, so it can have indiscriminate effects. And in the case of inflammation or in the case of MS, these reactive species will target all cells in the environment. And once it gets into the cell, it can cause havoc where it causes DNA damage, it causes organelle damage, and it causes mitochondrial dysfunction. So where the oxidative stress, for example, changes the balance or the membrane potential of mitochondria, and when the mitochondria is affected, it leads to the production of more oxidative stress. So it's kind of like they feed into each other, but they're also independent processes, but together they lead to cell death. I don't know if that what you were.
I just unmuted. Here, I'll show my face too. Actually, so I hit enter too fast. So do you think there's any good support for the notion that mitochondrial dysfunction in neurons might be underlying that or triggering that? Because some people think that, right? Do you think there's good evidence showing that?
So there are evidence showing, for example, changing in mitochondria shape, especially associated with aging. And when that happens, you do have, and with loss of the ability to undergo mitophagy, for example, those are all associated with increased stress to axons or to neurons. But I'm not sure whether, for example, when a person is diagnosed with relapse remitting MS when they're really young, whether mitochondrial stress is one of the factors that's causing the disease, rather than at that stage, perhaps it's more the inflammation or some other unknown cause that leads to oxidative stress, which leads to mitochondrial dysfunction. And that might be restored upon remyelination or remission. Whereas in progressive MS, and which goes hand in hand with aging often cases, then maybe the mitochondrial dysfunction it's a factor that we need to think about more in terms of treatment. Salma asks, does damaged mitochondria associated with MS have anything to do with mitochondrial DNA mutations? If so, does it mean that it can be inherited through a mother with MS? So mitochondria DNA mutations can be induced by stress and oxidative stress, so DNA will be damaged. But I don't know if this is inheritable. If anyone has a better idea than I do, please feel free to answer. No? Well, it's an open question, I guess. Good question. Len says, is there any particular type of exercise recommended for people with reduced mobility? I think either we or if Brian's here, they will be able to answer this better than I could.
The jury's out. What seems to be an emerging consensus is that we need moderate amount of exercise where one has some degree of sweat, if you like, for CNS outcomes. Whether you need that degree of exercise for reduction of fatigue, you know, that is it's unclear, you know. So we it's being debated. Intensity, dose, type, and for whatever for which outcome.
Marina asks, do you think we will be able to figure out ways to repair neurodegeneration in the future? Yeah, I hope we could. And I think the biggest challenge for us now is just we don't have a good model to study or repair neurodegeneration, and this is because the CNS is such an isolated tissue, where, for example, if you were to, use a drug, one of the challenges is, does it cross the blood-brain barrier, and does it hit to the specific site you want the drug to act in? So there are a lot of challenges in our way to figuring out how to repair neurodegeneration, but that's why we have active research programs, and the idea of repair is really an important, or it's being actively pursued more and more these days because we have such good treatments for the inflammation and demyelination. So really, yeah, the next step is to look at repair, but yeah, it will be challenging. We asked, do you know any antioxidants that goes into the CNS? So we work on one in our lab, indapamide. So that's a diuretic that's been shown in animal models that it can enter the CNS, and it has antioxidant potentials. So that's something we are pursuing. There are also others, for example, N-acetylcysteine. So that's a precursor to glutathione, which is a potent antioxidant in cells. That's also possible. There's also possibilities that it can enter the CNS, but again, at what concentration? So that's something that needs to be tested. And another way that's somewhat of an indirect way to upregulate antioxidants is if you can modulate the function of immune cells, for example, macrophages and microglia in CNS. If you can switch them to a phenotype where they upregulate antioxidants, so for example, something that dimethyl fumarate might be doing, then you get local increased levels of antioxidants. And that may also help with oxidative stress. So there's plenty of antioxidants out there. It's just that we have to test them in experiments and clinically to see if it works for MS. Do we know if unmyelinated fibers get damaged before the myelinated fiber upon CNS injury or disease with oxidative stress? That's a good question. And I think that's something might be interested in looking at. But if you were to think in terms of, I think you have to think of this as in terms of the sequences of events, right? This is really asking, do you have demyelination before you have axon damage? And my answer to that would be it probably happens at the same time. And in animal models, it probably happens at the same time with the entrance of immune cells, but I don't know what happens in MS. And that could be interesting because it will have implications on how you mitigate these responses. Lynn asks, are there any stem cell therapy for MS? There are. For example, you can get bone marrow transplant, and that seems to be good, or patients with bone marrow transplant seems to have, I wouldn't say cured, but they don't undergo relapse once they get the treatment. There's also explorations of mesenchymal stem cell therapy for MS, but I don't know if that's been as successful, but Canada for sure has one of the leading programs in hematopoietic stem cell transplant for MS.
I think we are done with the question and answer round. Thank you, Jeff. It was a wonderful talk, quite comprehensive, and I think our audience did well too. The discussion part was really good. So thank you so much. And the rest of the audience, all the post-event session are available on our website, so you can go there and check out. And we'll see you next Thursday. Bye-bye.
Thanks, everyone.